Final video here of chapter 12. We're going to look at some issues that can happen during the process of meiosis. Okay, so we talked a little bit about the genetic variability that occurs, and this is important because it keeps populations resistant to disease. It's kind of the whole idea behind natural selections. However, the making of these sex cells, if there's a mistake, we can get genetic diseases. So we've got a couple of different types. We can get a change in the number of actual chromosomes. We can have too many or too few. This is known as a non-disjunction. Or we could get a change in the structure of the chromosomes themselves. Okay. And so we've got that location where crossing over occurs, the chiasmata, and there can be deletions, duplications, inversions, and translocations during this process. And we'll look at all of these different types. So non-disjunction is when during metaphase 1 or metaphase 2, well, technically the anaphases, our chromosomes don't separate the way that they're supposed to. So our non-disjunction here in metaphase 1 is where our homologous chromosomes don't separate. And so in metaphase 2, we then have these 3 chromosomes here, 1 here. And so we have extra chromosomes down in these sex cells. And so that's going to be an issue. Okay? So we get unequal distribution of chromosomes during meiosis. So we've got few different types of things that can happen here. We can have monosomy, where we're 1 sex chromosome short. So in Turner syndrome, at least, they just have the 1 X chromosome. So if we look back here, these gametes would result in a monosomy, where there's only the 1 chromosome. These gametes on the right side would be a trisomy, where we've got an extra chromosome. Okay? And probably the most well-known example of trisomy is trisomy 21, Down syndrome, where the individuals with this genetic disease have an extra chromosome 21, and it causes all of those symptoms, those things that are recognizable with Down syndrome. Okay. We also see polyploidy in plants where they get these non-disjunctions, but typically it doesn't negatively impact the plants for whatever reason. So this shows our 2 types of non-disjunction. Here it is in meiosis 1 where the homologous chromosomes don't separate. And then here we've got a non-disjunction in meiosis 2 where the sister chromatids don't separate. And so here we've got a trisomy, here we've got a monosomy, and then in these ones, these are normal. So we've got a normal sperm cell down here and an egg cell that's got trisomy. And so here we've got this extra chromosome. Okay. Now, I will say that typically when there are trisomies or monosomies and fertilization does occur with those sex cells, that embryo is not going to be viable. Most of the time, this issue will result, it'll be fatal. So if you're familiar with miscarriages, a vast majority of miscarriages is due to some genetic issue. And sometimes that could be this non-disjunction that occurs where that zygote simply isn't going to survive long term because of the genetic defect that exists there. But there are some like trisomy 21, that Down syndrome, where that zygote will make it all the way to birth and they can live a full life. Okay. Other issues we have here, these are issues at the chiasmata. We talked about the crossing over where we get these exchanges of alleles with these homologous chromosomes. And this is normal. I'm not sure what happened there. Sorry about that. This is normal to get this crossing over, and it's totally fine for this to happen. But this has to be an equal exchange, as we see here bolded and in red, is we have to have the same genes being traded across these chromosomes. If not, there are going to be issues. Okay? And so we see these changes in chromosome structure with these unequal exchanges. Okay. And so we can get deletion, duplication, inversion, and translocation. And we'll look at each of these. Okay, so we get a deletion where a piece of a chromosome just breaks off and it doesn't go and reconnect to some other chromosome. It's just gone. And so you're missing entire genes from that chromosome and you're going to have some issues genetically. Okay, with Fragile X syndrome, a piece of the X chromosome breaks off. Males are affected most because males only have one X chromosome. And symptoms, we've got this mental defects, unusually large testes, protruding ears, because of these missing genes from that X chromosome. Okay. Duplication. Okay, in a duplication, we're going to have a piece break off of one chromosome and it's going to end up on that homolog. So we get a deletion in one of the chromosomes and then an extra set of those genes in another chromosome. Okay, and so this is going to cause issues. An inversion is where a piece is going to break off and then it's going to turn around. So it's going to be in the incorrect order. Now, at face value, this doesn't seem like it's going to be an issue, but when we go through our processes of transcription and then translation, having these genes backwards is going to cause some issues in those cells. And then we've got a translocation where it breaks off of one chromosome and ends up on a totally different homologous, non-homologous chromosome. So we get a piece that breaks off of, let's say, chromosome 1. And then it ends up on chromosome 6. So we've got these genes over here where they're not supposed to be. Then we're missing those genes in the area that they are supposed to be. And so we've got various genetic diseases that can result from this. But all of these issues as well, similar to our non-disjunctions, if we get these chromosomal issues, it's very possible that if fertilization does occur with this gamete, it's going to create a non-viable embryo. And so it simply won't be able to make it up through birth. Okay. And I believe that's our last one. So those are our issues with meiosis, and that is the end of chapter 12.